The overall goal of this procedure is to expand patient-derived esophageal epithelial cells from endoscopic biopsies to study esophageal diseases or to create a 3D esophageal scaffold for downstream modeling or esophageal tissue replacement.
This method can be used to better understand the role of esophageal epithelial cells in esophageal disease. They can also be used to seed 3D scaffolds for modeling or implantation. The advantage to this technique is it allows us to generate patient-specific epithelial cells from a very small piece of tissue without the need for any genetic or permanent modifications. The implications of this technique extend towards understanding the pathogenesis of eosinophilic esophagitis, as well as developing treatments for esophageal atresia and eosinophilic esophagitis. Generally, individuals new to this technique will struggle with the ability to completely remove the feeder cells prior to harvesting human esophageal epithelial cells in order to maintain purity. Demonstrating this technique will be Christopher Foster, a technician from our laboratory.
At least 3 days before the patient samples are obtained, plate the 3T3 cells in 3T3 culture medium in tissue-treated T75 flasks. On the day of the experiment, just prior to obtaining the samples, detach the 3T3 cells with 5 ml of 0.25% trypsin EDTA per flask for about 5 minutes at 37 degrees Celsius. When most of the cells are floating, add 5 ml of 3T3 medium to stop the reaction and transfer the cell suspension into a 15 ml conical tube for centrifugation.